Okay, let's get started. Welcome back. Let's take a minute or two to shake the rust off our brains and remember where we are and where we're going. We're making our way through the middle theme of the course, which is open challenges in the field. Hopefully you've all been convinced by now that evolutionary robotics is obviously the way to eventually do true AI. However, it's a relatively young field and there are a number of big open and unanswered questions. We'll get to we'll start in on lecture 15 today, which is the first of a 6-part series in arguably the biggest open problem in the field, which is you evolve some amazing robot in simulation. You go to the time and expense and effort to build a physical copy of that evolved simulated robot, and the physical robot does not do what you saw in simulation. So how do we go about trying to cross the reality gap? We're going to look at 6 lectures that are organized in time. This is the first attempt, the radical envelope of noise hypothesis, which we'll look at today, which dates back to the early 90s and the beginnings of the field, up to the 3D printing of 1D robots, which is a research paper that was published last year, and at the moment is sort of the best answer we have in the field to crossing the reality gap. But again, as you can see from this list and the most recent of these attempts, there are probably other solutions out there waiting to be found. So we'll spend the next couple of weeks investigating some of those attempts. Before we do, we will back up to lecture 14 when we were looking at 2 different algorithms. We introduced the first of the 2 algorithms 2 weeks ago, the NEAT algorithm. And the NEAT algorithm is a solution to an open challenge or an open problem. What is that open problem? What is the problem to which NEAT is a possible solution? Yes?
Crossing over and evolving neural networks.
Exactly. So crossing to evolved neural networks, or trying to combine 2 good but not optimal neural networks is a huge open problem. Again, not just in our field, but in AI in general. You train 2 different neural networks and they might capture parts of the solutions to some of the subproblems. So if the problem you're trying to solve is modular in some way, and you train a neural network to solve that problem, different parts of the inner structure of the neural network might learn to solve different modules or subproblems. But other subproblems that given neural network may not do such a good job at. Another neural network might solve a separate subset of the subproblems. And if you can just pull out the 2 working parts of the 2 neural networks and combine them in just the right way, you would have a new neural network that does better than either parent. This works in practice. We're living proof of that. Sexual recombination in nature does work, and it can, under the right circumstances, bring together the best of both worlds, but it is a difficult thing to get right. And NEAT, although it's been around for 15 years, is still sort of the best solution we've come up with to that problem. We'll talk about HyperNEAT in a moment, but before we do, let's pause and switch back to the assignments. Hopefully everybody has now completed assignment 8, where we've discarded the very simple scissor bot and you've replaced it with the more complex quadruped. Undergraduates, you're now moving on to assignment 9, which I won't bring up here, but assignment 9 is going back to this sequence of creating more high-powered evolutionary algorithms. And in assignment 9, you're going to be implementing the most common evolutionary algorithm, which is the genetic algorithm. Next week, in assignment 10, you will tackle the 10th and final assignment. We'll talk about that next week. The end is in sight. Any questions about the assignments? Grad students, any questions about weekly reports, final projects? Think everything's coming along well? Okay, so back to NEAT and HyperNEAT. So as we just mentioned, we're trying to solve this problem, which is otherwise known as the competing conventions problem. You might have 2 neural networks that are good at different parts of the problem, but those different sub-solutions to subproblems may be in different positions in the neural network, or in the 2 neural networks. How do we ever know which are the corresponding pieces in 2 separate neural networks? The idea in NEAT is to use the idea of a common ancestor. So if you have an ancestral neural network and there's some part of that neural network that was doing okay at solving some subproblem, that neural network, maybe through mutation, produced a whole bunch of children and grandchildren and great-grandchildren. All of them may have certain parts within them, and those parts, like A, for example, or B, they may be in different positions in the descendants, but if you can trace those parts back to the same part in their common ancestor, they're likely to do similar things in the descendants because they descended from the same component in the ancestor. That's the intuition underlying the NEAT algorithm. That's the intuition. How do we turn that into an algorithm? In NEAT, we encode in the genotype two lists, one list for the neurons and the second list for the synapses. And every neuron and every synapse has associated with it this innovation number. The first time that a neuron is added to a list or a synapse is added to a list, when we add a new piece of neural network, we assign it a new value. And if that neural network survives and produces children, that neuron or synapse that was added in the ancestor, that corresponding part in the descendant has the same innovation number. So if we take two neural networks, let's go to crossover here. We take two neural networks down here, and we see, for example, let's see if we can find one here. This one here, the synapse that connects neuron 4 to neuron 5 in this neural network, and the synapse that connects neuron 4 to 5 in that neural network. In this case, they both have an innovation number of 6. So they descended from a same common ancestor in which that synapse, the one that connected neuron 4 to neuron 5 in the ancestor, had an innovation number of 6. We take all of the synapses, and this is just the list of synapses. We're not looking at the list of neurons here. We line the list of synapses from parent 1 up against the list of synapses from parent 2. And we then cross over, we select one synapse from each of the parents. When there are corresponding alleles, and we're going to borrow this terminology from evolutionary biology, an allele indicates that there's two flavors or two options for the same gene, or in this case for the same synapse. We pick one or the other from parent 1 or parent 2. In some cases, there's a synapse that exists in a particular parent that has no corresponding allele in the other parent. We copy that into the offspring and continue. And if we use the NEAT algorithm, we tend to obtain children that have architectures which look similar to both parents but not identical. And we ensure that we're copying into the child one and only one copy of corresponding structures in the two parents. That's the idea. Another open problem in the field is not just how to combine material from two different neural networks, but how to actually alter the architecture of neural networks themselves. The deep learning revolution showed us how to train synaptic weights for a neural network with a fixed architecture. But the moment you make a change to that architecture, typically if you're using a learning algorithm, you kind of have to start from scratch all over again. So a big open problem, again, not just in our field, but in AI and deep learning in general, is how do we alter not just the synaptic weights, but also the underlying architecture of the neural network itself. And NEAT is a pretty good contender. It tends to work pretty well. Okay. That's the NEAT algorithm, which is to solve this competing conventions problem. We're now going to look at the HyperNEAT algorithm, which uses NEAT as just one component of its operation. HyperNEAT is actually designed to solve a slightly different problem, which is that if you typically look at things that you're evolving, there tends to not be very much structure. So if you think about your own assignments at the moment when you're evolving neural networks, now for the quadruped, forget how many synapses there are in the quadruped. There's a couple dozen. If you looked at the evolving weights, they're not really related to one another. They're all sort of independent, different values, and evolution through mutation is sort of tinkering with those values. If you crack open a biological organism and look inside, it doesn't look like a random agglomeration of parts. You tend to see regular structure inside the anatomy, and also the nervous systems of organisms. So another open problem in the field is how do we bias evolution towards regular structures rather than just random assortments, random aggregations of values and structures. So here's a little cartoon of a biological organism here. What do we mean by regular structure? Well, there's lots of regularity you can see in biological organisms. What kinds of regularity that you see in insects, for example? You see symmetry, right? So in most higher animals, you tend to see bilateral symmetry. The left side of the body looks like the right side of the body. If you look inside the neural network of your virtual quadruped, and you were to look at the left side of the neural network and the right side of the neural network, or the side of the network that connects left sensors to left motors and right sensors to right motors, tends not to be bilaterally symmetric, right? What are some other forms of regularity you see in biological organisms?
Recurring structures like multiple legs.
Absolutely, recurring structures. So the little cartoon here of an insect is on purpose. Particularly insects, you tend to see segmentation or repetition of segments or modules. We're back to modularity again. Modularity is a form of regularity. What else?
The same organ is adapted to be, like, have a different function, but with the same kind of base genetic coding.
Exactly. So you tend to see specialization, right? So every cell in your body has exactly the same copy of your DNA, but the subset of genes that are expressed in every given cell might be slightly different. So the way that natural evolution tends to proceed is to have sort of a basic template and then turn on and off different parts of that template to specialize certain structures at certain places and at certain times in an organism. And so this very mammalian looking heart that's in the insect here is just there's a reminder that in the same organism you can get symmetry, you can get repetition, but you can also get specialization. You can get organs and organelles and certain structures if needed. Okay, so how do we go about designing an evolutionary algorithm that doesn't randomly place things in different positions, but instead tends to find solutions that have regular structure throughout them? And the idea here, the intuition is obviously regularity is more likely to give you the function that you're after than a random collection of parts. Imagine an asymmetric organism or an organism in which it's very unlikely to see segments or repetition. You can imagine the vast majority of body plans you can imagine actually don't work very well. The ones that we see on Earth tend to all be regular in different ways. It tends to be by a lot of symmetry, repetition, specialization of structure and function, and so on. So the HyperNEAT algorithm, which we're going to talk about now, biases search towards genotypes, which you'll see on the left and we'll talk about in a moment, that tend to produce phenotypes that have regular structure. Okay, so what is the genotype in HyperNEAT? Well, HyperNEAT is going to evolve populations of neural networks, so not that different from what we've seen before. But the particular networks that are evolved by HyperNEAT are a particular form of neural network that we haven't seen before, and these are known as CPPNs, or compositional pattern producing networks, not to be confused with CTRNNs, which we saw when we were talking about minimal cognition, continuous time recurrent neural networks. CPPNs are a very different animal, and we'll see why in a moment. Let's start with the C, or the composition. So here's a CPPN over here. Again, it looks not unfamiliar to you, hopefully at this point. It has an input layer up here, and that input layer is connected by synapses, represented by the arrows, to the circles that you see, which are representing neurons. And in this case, we have just a single output neuron here. You'll notice in a CPPN that there are little functions that are drawn inside each neuron. What are those functions? Remember back to our discussion about neural networks towards the beginning of the course. We talked about all the sort of building blocks that make up a neural network. What are they?
Activation functions.
They're activation functions, right? So remember, if we take any given neuron, like this one here, that neuron has two incoming synapses. We multiply the value of the incoming of the source neuron by the incoming synaptic weight plus the value of the source, this source neuron times the weight of this synapse, which gives us a raw value that's arriving at the neuron. We then push that raw value through an activation function, and the value that comes out the other side of the activation function is the actual value of the neuron, and that actual value flows out along any outgoing synapses from the neuron. What do you think the little x and y axes are inside these activation function visualizations? What's x and y here?
Input and output.
Exactly. So you can think about these little inset axes where the x value is the raw input and the y value is the activated value. We're pushing that x value through the activation function, and the activation function gives us back the y value, which is the value that flows out along outgoing synapses. Yes?
Do biological neurons have activation functions like this? Because if they did, it would suggest that they'd be able to do things like calculate the side function by themselves.
That's a good question. So do biological neurons have an analog of the activation function? We'll turn that question around. Activation functions were modeled on the function of biological neurons. Biological neurons have lots of differences compared to the traditional neurons you see here. One of the big differences is that they don't deal with floating point values arriving at their inputs. They deal with electrical spikes that are arriving. So biological neurons actually talk to one another through spikes of electrical activity, and the time interval between these arriving spikes is what influences what the neuron actually does on its outgoing synapses. And what any biological neuron does is actually very complicated, and in a lot of cases we don't actually know. But it is a function of the incoming spike trains, or the series of electrical spikes that are arriving at a neuron along its incoming synapses. So there is a transformation, and it is definitely true that biological neurons transform their incoming spike trains in different ways. That's sort of where the similarity ends. So as you can see in the cartoon here, the thing that distinguishes CPPNs from all the other neural networks you've probably seen so far is each individual neuron in a CPPN can have a different activation function. And if you think about the values that are flowing down from the input layer to the output layer, they are going through one function and then going through another function. So the values are transformed by one function and then transformed by another function and possibly another function and so on. Back to high school math again. You've probably seen this before as the composition of functions, right? You all remember f of g of x. So you can imagine x, if this is a vector, as our vector of incoming values. G is one of the first neurons that's encountered. F is the next one that's encountered as the values are flowing down, and so on. So what a CPPN is actually doing is composing functions together. Okay. Why CPPNs do that, we haven't talked about yet. What they're designed to do, as the rest of the name implies, is to produce regular patterns, pattern-producing networks. How do CPPNs do this? The second distinguishing feature of CPPNs is that the input layer is quite special. The input layer receives input coordinates from particular points in particular spatial positions in the phenotype. So let's look at a very, very simple phenotype. Let's not look at cartoons, insects, or neural networks, or robots. We're going to take a simple two-dimensional plane, and our CPPN is going to paint a phenotype into this two-dimensional plane. We've got our x and our y, and for simplicity's sake, let's assume that both x and y range between 0 and 1. We're going to visit regular points in this two-dimensional plane, and at each particular point, we're going to have a coordinate, in this case 0, 0. So those two numbers, 0, 0, become the input to the CPPN, which is painting this phenotype. We then go to the next point, which is 0.1, 0. That becomes two new values that are supplied to the same CPPN, and it is going to paint something onto that position there. We're going to visit the next position, the next position, the next position, and so on. So far, so good? Let's create a very simple CPPN. We're going to have, obviously, our two input neurons here, X and Y. And we're going to have one single output here, which is, we're going to have a single output, and we'll put a very simple activation function in here, which is the identity function. Whatever the incoming raw sum is, that's the value that leaves the output neuron. So far, so good? Let's connect with just a single synapse, the X coordinate to the output value. And again, in the spirit of trying to create the simplest possible CPPN we can, let's give it a synaptic weight of 1. Okay, so let's come back to 0, 0. We supply 0, 0 to the input layer. You're all neural network experts now. What arrives at the single output neuron? What's the single value we get back when we supply 0, 0 at the input layer?
0.
0. So 0 times 1 is 0. X, in this case, is 0. Because we're using the identity function, y is our output. So we get 0 down here. And let's treat this outgoing value as a color. It's not a color because we have a single number. So we'll treat it as a grayscale value. This value, as we can see in this picture here, is always going to be between 0 and 1. So we'll treat 0 as black and 1 as white, and anything in between as gray. I don't have black chalk, I have white chalk, so I'm going to draw black here. Let's now visit 0.1, 0. Supply that to the input layer. What do we get at the output layer now? 0.1, right? So we've got 0.1 here, 0 here, 0.1 times 1 is 0.1. Push this through the activation function. We get 0.1 here, so we get a very light gray at this position. What amount of gray do we get at 0.2, 0? Slightly darker, so we get a value of 0.2 here. If we keep going, we get darker and darker and darker, or lighter and lighter and lighter. It doesn't really matter for our purposes, right? So we've got this CPPN, and we're querying the CPPN multiple times as we visit different positions in the phenotype. And I know it seems strange that our phenotype is a two-dimensional plane for the moment, but we'll get to robots in a few minutes. What happens at this value? This value is now 0, 0.1.
What's the weight for the y?
There is no weight there. There's no synapse connecting y to 0. So what do we get at this position?
It's going to be the same.
It's the same, right? 0.1, 0, 0, 0.1 gives us 0 at the output layer. So we have black here. What happens as we travel from left to right in this row of the two-dimensional plane?
It will be identical.
It'll be identical, right? So we're going to get, again, a gradient that gets lighter and lighter as we travel from left to right. What happens at the third row? Same thing, right? Okay. I'm not the most accomplished artist, but I think you get the idea. Tell me about this phenotype. Is it a random collection of grayscale values? Is it white noise? Definitely not. Now, of course, I cooked this example, but the beautiful thing about CPPNs, and if you want to play around with this in Python, as you can see, it's not too difficult to do, you can create a random CPPN, a random collection of inputs and outputs and hidden neurons and activation functions and synaptic weights, put them all together in a random arrangement, and you tend to get non-random phenotypes. Which particular one you get, obviously, is random, but you tend to see regular pattern here. We've got a particular regular pattern here, which in this case is gradient. As we move from left to right, we get lighter and lighter. There is some symmetry here. The top half is the same as the bottom half. What about this CPPN? What pattern does this particular CPPN paint in the same two-dimensional plane?
It's the same, but 90 degrees, so.
Exactly. So now we're going to get a lighter and lighter gradient as we get higher and higher.
Does this system allow for negative numbers?
Depends. We could. At the moment, if we're treating our phenotype as a field of values between 0 and 1, then we may want to clamp our output value to give us back a range between 0 and 1. So we might cheat by putting an activation function in our output neuron that we know guarantees to give us back a value between 0 and 1. If we do that, then we can relax and allow everything inside the neural network to give us any range, have any range that we want. Let's play the inverse problem. Let's say we want a phenotype that has more and more white as we travel towards the upper right and more dark as we travel towards the bottom left. What CPPN would produce this phenotype?
X plus Y.
And we could put something in here to clamp between 0 and 1, but we could also just have the synaptic weights, and that would give us, again, another gradient. What happens if we're interested in a different kind of regular pattern, like again we see in the cartoon over here, where maybe we want to have oscillations in gray as we travel from left to right? What do we need in that case? How do we need to alter our CPPN to produce bands of white as we move from left to right?
Some kind of sine function?
Exactly. So again, if you look at the cartoon here, you'll see in CPPNs, one of the available activation functions is a sinusoidal or more or less sinusoidal pattern. So in CPPNs, the evolutionary algorithm is not just setting the synaptic weights, but it is also choosing and altering the type of function that sits inside of each neuron. Okay. Let's talk now about the resolution of the phenotype. So in the cartoon here, I was skipping from point to point with a distance of 0.1. Does it matter? Let's go back to the original CPPN we were looking at here. What would happen if I traveled instead 0.1, I traveled 0.01 or 0.001? How would that alter the image that we get here?
It would just be higher resolution.
It would just be higher resolution. One of the nice things about CPPNs, which again we'll see in a moment, is the same CPPN can be queried multiple times to produce phenotypes at different resolutions. And at lower or higher resolution, they have more or less the same structure. So this one still produces this gradient of white going from left to right, but the resolution of that gradient is just depending on our choice of how densely we query the CPPN across this two-dimensional space. So far, so good? Okay, so this is obviously a very abstract example. We could use CPPNs to produce pretty pictures in a two-dimensional space. And you can actually play around with this if you open the website PicBreeder. PicBreeder was an original website meant to sort of show off what HyperNEAT can do. forget what it is. If you just Google PicBreeder, you should be able to find it. As you might be able to infer from the name, if you go to the PicBreeder website, it will show you 16 random two-dimensional images. You click on 2 or 3 of them that you like, and then you click on on the next button. What do you think happens on the next page of the website? You were shown 16 random pictures. You chose 2 or 3 of them. What happens next?
It crosses them.
Possibly. As we know, crossover is a little bit tricky. HyperNEAT does have NEAT built into it, so there is some crossover. What else is likely to happen from one page to the next?
Would it maybe show another set of pictures that have characteristics of each of those pictures that you selected?
Exactly. So we're going to take those 16. If I chose 3 of those 16, it's going to delete the other 13, the ones that I didn't choose. And then it's going to make randomly modified copies of the 3 survivors. And it might do that through mutation or crossover. What happens if you mutate a CPPN? Let's take this CPPN. We know what phenotype it produces. Let's make a relatively simple mutation. Let's just lower the synaptic weight. This is the child CPPN. What pattern does this CPPN produce?
It's going to be zoomed in. I guess like it's only ever going to reach 0.5 as the highest value.
We're going to get the same gradient, but it'll just lighten at a lower rate, right? We'll just get a darker left to right grayscale gradient. So you can, again, play around with this with pen and paper or code this up in Python and play around with it in Matplotlib. It's a lot of fun to play around with CPPNs because if you create even simple ones and alter simple ones, you get interesting patterns. You get a feel for how genetic alterations to a CPPN genotype alter the phenotype, but the new phenotypes tend to also have regular patterns. It's very, very difficult to create a CPPN that produces random grayscale values across your 2-dimensional image. So far, so good? Question?
So does it ever evolve the activation functions in terms of cutting out neurons or just giving around?
Good question, right? So does it ever alter the activation functions or cut out neurons or add neurons or delete neurons or delete synapses, add synapses? Yes to all of the above, because in HyperNEAT you have a population of CPPNs, and these CPPNs are evolved using NEAT. So the actual neural networks that are being altered by the NEAT algorithm are CPPNs. That's what makes HyperNEAT HyperNEAT. We evolve populations of CPPNs using NEAT. So in HyperNEAT, you often start with very simple CPPNs like this, and then as evolution proceeds, NEAT adds neurons and synapses and alters activation functions to create more complex phenotypes, but phenotypes that still exhibit regular structure.
So does the neuron average all the incoming stuff into one thing that it plugs into the function, or does each one have its own variable in the function?
Good question. So in this example here, we're obviously throwing away one of our inputs. In this example here, you can see that there are 2 input neurons that are not quite averaging. Both neurons here actually do different things, but they're combining the input layer in different ways. But that doesn't necessarily need to be the case. Imagine that this particular neuron is not there.
Yep.
And synapses flow from here to here and from here to here. You can have arbitrary compositions of functions. When you do that, you're composing functions, and those functions actually alter your coordinate system. So what CPPNs are really doing is composing coordinate transformations. And if you compose coordinate transforms, what you tend to get are very pretty pictures. You get regularity within regularity within regularity. It's not quite fractals, but it's heading in that direction. So far, so good?
So would you have to, the initial population of these networks, do you have to give a predefined set of functions, basic functions to start from, and then the evolution changes coefficients?
There's a fixed set of activation functions that come preloaded with HyperNEAT. You get things like the sine and cos function, which give you oscillations like we saw before. You get the identity function, and the identity function, basically, as you saw here, gives you sort of gradients across the coordinate system. There's Gaussians that are built in, absolute functions. As you can see in the example on the right here, the Gaussian and the absolute function tend to give you symmetry, right? As x increases, it can also cause y to start to decrease. So you can start to get symmetries as well through the absolute and Gaussian functions. As you compose these together, you tend to get repetitions of things. Within any one repetition, you might get symmetry. Within that symmetry, you might get a different repetition. Within there, you might get some specialization, and so on and so forth. You're composing coordinate transforms. So it gets complicated. The phenotypes get more complicated as you make the genotype more complicated. But you're biasing evolution to always give you back a phenotype that has some amount of regular structure within it. So far, so good? Okay, so let's expand the reach of our CPPN now. We have a simple CPPN that is limited to producing grayscale values inside of a two-dimensional image. Let's enrich our CPPN so now it can paint color images. So the one on the right is still two-dimensional, but it's in color. How would you alter the architecture of a CPPN so that it can paint in color rather than grayscale? Yes?
Three outputs.
Exactly. So now we're going to fix the output layer to have 3 output neurons, and the value arriving at the first one is the amount of red, the value arriving at the second one is the amount of green, and the value arriving at the third one is the amount of red. So now when we visit each individual two-dimensional coordinate, we're going to get back a triplet of values, and we can treat those as a color, and now we're drawing in color. Let's be a little bit more ambitious. Instead of painting inside a two-dimensional space, I want to paint colorized voxels, or three-dimensional pixels, inside of a three-dimensional space. How do I alter the CPPN so now it's producing regular patterns in a 3D space rather than a 2D space?
Give it a third input.
Exactly. Now we fix the output layer to have the input layer to have 3 values. We visit each point inside a three-dimensional volume, and the output layer is now painting voxels inside a three-dimensional cage. You've actually already seen or heard this idea in passing in this course. Where did you hear this idea before? Painting voxels inside of an empty cage. Where have you seen collections of three-dimensional voxels before with different colors?
The lecture on soft robotics.
Exactly. So your guest lecturer, Sam Kriegman, my PhD student, uses HyperNEAT to paint 3D voxels inside a space, and those voxels are then placed inside a physics engine, a soft physics engine, and those voxels, the color then represents the material properties of those voxels. So what Sam is really doing is not painting pretty pictures inside a three-dimensional space, but building a three-dimensional shape or evolving three-dimensional shapes using HyperNEAT, and those three-dimensional shapes become 3D soft robots. So we're back to robots. Okay, let's come back to pictures for a moment. Instead of producing 2D or 3D images, I want to produce a cool movie that I want to upload to YouTube. How do I alter CPPNs to produce animations rather than a fixed image?
4 inputs.
Now I add T or F, where F is maybe going to be an integer, which is the particular frame in the movie that I'm producing, right? In this case, we would now be producing a 3D movie over time. The nice thing about CPPNs, which again we'll see in a moment, is that they can paint regular structures inside an arbitrary dimensional space, and they can alter that pattern over time if we wish. Okay, as you can imagine, we're going to see HyperNEAT applied to evolve robots in a moment. You can go to PicBreeder and use it to evolve pretty pictures. What else might you be able to use HyperNEAT to do?
Involve robot shape.
We could evolve robots' shape, and again, we're going to see that when we talk about soft robotics towards the end of the course.
Use it to evolve gates.
Could use it to evolve gates. That we will also see in a few minutes. I know robots are awesome, but what else can we do with HyperNEAT other than evolve robots? We can evolve images, we can evolve movies, we can evolve robots. Evolutionary algorithms are kind of agnostic to the phenotype, right? The evolutionary algorithm doesn't care whether it's a robot or an image. It's just evolving regular patterns inside some arbitrarily dimensional space. If we can use HyperNEAT to evolve the 3D shape of robots, what else might we be able to evolve?
A controller.
A controller, possibly. Let's think broader here.
Like three-dimensional shapes of objects, like arrows, like planes.
Absolutely, right? So I think at the beginning of the course, I was telling you about evolving the nose cones for aircraft. We can evolve arbitrary shapes now in three-dimensional space. Autodesk, the company that produces state-of-the-art CAD software, up until relatively recently, if you used CAD software, you made the three-dimensional shape. Autodesk has now produced a product called Dreamweaver, which basically uses something like a CPPN. Not quite sure, because again, it's a trade secret, but they're using some kind of evolutionary algorithm in the CAD software, where now instead of you designing the shape by hand, you instead specify the function that the shape should support. And Dreamweaver gives you back a shape that confers that function. What else? Robots, 3D shapes. Any musically inclined people here? How would you evolve music using HyperNEAT? I don't know if it would be appealing music, but you could imagine trying it.
You could have like an output variable instead of being like a color scale, would be frequencies, and time would still line up, I guess, with like timing and so on.
That's a good idea, absolutely. So we could supply frequency and time, or I guess we would have frequency at the output layer.
You also need volume too.
Volume, okay.
And like timbre, or like what kind of sounds.
Okay. Remember that we're probably going to have to paint a regular pattern in space and time. Definitely time, because we're talking about music. What is the space in this case? Let's leave our time input neuron here.
Would it be instruments?
Possibly. We could print music onto sheet music, where we could have each row represent, again, a height in HyperNEAT, and each horizontal position here would be time, and the output might be a quarter note, a half note, a whole note, timbre, which instrument, and so on. For those that are musically inclined, I will leave that as an exercise for the reader. The point here is, again, that HyperNEAT can produce regular patterns, and regular patterns are useful in lots of human domains: robotics, music, control policies, architecture. It's been used for architecture. It's now being used for drug design. Instead of placing color at particular positions, maybe you want to place atoms or molecules. You can use it for drug design, being used by pharmaceutical companies as a possible way to automate drug design. Lots of uses for HyperNEAT, but as you can imagine, we are going to use this for evolving robots. Okay. How are we going to do that? We're going to do something which initially seems a little bit strange. Instead of painting color onto pixels, we're going to paint weights onto synapses. So we're going to construct yet another neural network. This one, as you can see, is a neural network that is embedded in three-dimensional space. The front face of this cube contains all of the sensor neurons. the middle cut through the cube contains 5 by 4 or 20 hidden neurons. And the back layer contains all of our motor neurons. We're going to look at a quadruped in a moment. So in this case, we have a quadruped with 4 legs, and we're going to place a proprioceptive sensor at each of the forward and back joints in the 4 hips. We're going to place a second proprioceptive sensor in the in and out joint at the hip, a third proprioceptive sensor in the knee, and finally a fourth touch sensor in the foot. So for each of the 4 legs, we have 3 proprioceptive sensors and 1 touch sensor. So we've got a total of 16 sensors, and then we have a 17th, 18th, and 19th sensor in the main body, which gives us yaw, pitch, and roll. Yaw, pitch, and roll. And then finally, a sine wave. What is the neuron here producing a sine wave? What type of neuron is that? Anybody remember?
The central clock. I forget.
The central pattern generator, right? So we have this 20th neuron here, which is just outputting a constant sinusoidal pattern. Not too unfamiliar, right? So we have all 20 input neurons here, and each input neuron here sends an outgoing synapse to each of the 20 hidden neurons at the next layer. Each hidden neuron sends a synapse to the 1, 2, 3 times 1, 2, 3, 4, 12 motor neurons. Each row in the back here corresponds to one of the 4 legs, and each leg has 3 motors associated with it: forward and back, input and out, and the knee joint. Right? Okay, we could do what we always do, which is just take all of those synapses, put them in one long vector, and evolve the floating point values in those vectors. But that will give us back a collection of synaptic weights in which there's no sort of rhyme or reason, right? Everything is independent of everything else. Let's instead take a CPPN and paint synaptic weights onto this network. If we do, and if CPPNs do what they're supposed to do, we should get regular patterns of synaptic weights across this 3-dimensional volume. So far, so good? Okay, how do we do that? What's our input and output layer for our CPPNs if we want the CPPN to paint regular patterns of synaptic weights across this neural network? What are the neurons at our input layer? So we know that this neural network is embedded in a 3-dimensional space. So what do we probably need at the input layer?
X, Y, Z.
We need X, Y, Z, right? So back to X, Y, and Z. What do we need at the output layer?
The 12 motor neurons.
Not quite. We're going to paint synaptic weights. We're going to paint a weight onto each synapse in this neural network. And of course, the synapses aren't drawn here, but imagine a whole bunch of synapses going from the leftmost plate to the middle plate, and then a whole bunch of synapses going from the middle plate projecting onto the back plate. We got hundreds of synapses.
Do you need an output for every synapse, or do you only need it for synapses that are going to have the same?
Do we need an output for every synapse, or do we need just one output? We probably have hundreds of synapses in this case. It would be problematic if we had a neural network with several hundred outputs, right? So the beauty of a CPPN is in this case, like most cases, we just need one output neuron, W.
But what if it has a different weight depending on which neuron it's connected to.
Ah, okay. Well, let's see what happens with this CPPN. We won't worry about what's in here. What we're going to do is we're going to visit each synapse. So let's, for example, visit the synapse that connects this input neuron to this hidden neuron here. We see that this synapse exists in a three-dimensional space. The synapse starts here and goes here. So for convenience, let's take the center of this arrow as the x, y, and z coordinate of that synapse. So we calculate the x, y, z position of the center of that arrow. We supply that to the input layer of the CPPN, and the single floating point value that arrives at the output layer becomes the weight of that synapse. Now we go to the synapse that connects this input neuron to this hidden neuron, which obviously has a different arrow and a different xyz as its midpoint. We take the xyz coordinate of that synapse, supply it to the input layer, and probably we'll get a different W.
So think about the one on the far right and the far left, and then what if you're trying to find the weights of like crossing, and then they have the same midpoint.
Excellent observation, right? So we're close here. We've at least simplified our genotype so we don't have one output neuron for every synapse. We have a relatively compact CPPN, which in this case can paint different W's onto several hundred synapses. But unfortunately, our CPPN is too simple. As you've pointed out, imagine the synapse that connects this input neuron to this hidden neuron, and consider this synapse that connects this input neuron to this hidden neuron. They both cross over at exactly the same point. Those two synapses have exactly the same XYZ for their midpoint. What can you tell me about the weights of those two synapses? They're the same, right? So we're sort of hobbling our CPPN. It cannot produce different weights for synapses that cross at their midpoint. And given this embedding, this way of embedding this neural network in three-dimensional space, we can't get around that. How can we solve this problem?
Instead of using the midpoint, you could use something closer to the outgoing.
We could, right? Instead of the midpoint, maybe something a little bit closer to the target neuron or a little bit closer to the source neuron, and hope that we don't have anything that overlaps. Could do that.
Embed this in one dimension?
You're getting closer, right? So our neural network is embedded in three-dimensional space. So we're painting a regular pattern of weights inside a three-dimensional space. But nobody said that the dimensionality of the CPPN has to be the same as the dimensionality of the neural network.
So another for like the ID of the neuron.
Okay, now we're getting there. So now we're about to leave three-dimensional space, which means I can't draw anymore. So turn on your mental simulators here for greater than three-dimensional space. We're going to instead embed our CPPN in six-dimensional space. All right? So that means we need to have 6 input neurons. We've got X, Y, and Z here. The first 3 are going to be the X, Y, and Z of the source neuron. And the 4th, 5th, and 6th are going to be the X, Y, and Z coordinate of the target neuron. So this 6-dimensional coordinate system will uniquely identify every synapse that exists in this neural network. You're right, we probably could have assigned an ID and gotten away with 4. But in this case, they went to 6. Question?
So what happens when the source and I guess sync neurons are switched?
Ah,
And
good question.
is it a different value for what should be the same?
Possibly. So I'm not sure if they described this in the paper, but for our purposes, let's assume that the hidden layer has recurrent connections. Let's assume that there's a synapse that goes from this neuron to this neuron, and another synapse that goes from this neuron to this neuron. Do those two neurons that go from I to J, neuron I to J, and then another synapse that goes from neuron J to neuron I, do they have the same weights?
They would have to.
I know it's early in the morning, but you're going to have to do this in 6-dimensional space.
Yeah, I want to know.
They don't have this. They don't necessarily have the same W because their address in 6-dimensional space is different, right? The neuron going from I to J has xi, yi, zi, xj, yj, zj. That's the address for the synapse going from I to J. And the address for the neuron, the synapse going from J to I, has address xj, yj, zj, xi, yi, zi. So our CPPN that exists in 6-dimensional space has quite a bit of freedom. It can independently label the weights of synapses, even if they're recurrent. I goes to J and J goes to I. So far, so good? Okay. That's fine. That's all well and good, but you may have noticed this looks a little bit strange now. We have populations of these neural networks, CPPNs, being evolved using NEAT. So we have populations of neural networks that are being evolved. Each one CPPN in the population creates another neural network, which is the one that controls the robot. Why the heck would we go to all that effort to evolve populations of neural networks, each of which produces a neural network, each of which controls the robot?
If we want a robot to be capable of multiple tasks.
Possibly. The one thing we do know is that HyperNEAT, as we've seen, will produce regular patterns in space. So by evolving CPPNs, we know that in these neural networks, the ones that control the robots, we're going to have regular patterns of synaptic weights across this 3-dimensional volume. If you go and Google HyperNEAT, and I think if you do it in Google Images, there are actually some 3D plots that show distributions of synaptic weights through the 3D space of a neural network. You can actually see a visualization of it. That's great. The missing piece is, as roboticists, who cares? Is that going to tend to help us evolve behaviors for our robots? All right, that's the missing piece, and we'll touch on that in a moment. There's my little shorthand for the genotype that produces this phenotype, right? Okay. As you start to build up an intuition for how HyperNEAT works, as some of you mentioned, there are alternate encodings in different dimensions that you can also imagine, and they sort of have their pros and cons. It's, again, something that's kind of fun to play around with. Question?
This reminds me a little bit of variational autoencoders.
Okay. They share something in common in that there's definitely a compact encoding that generates it generates a pattern, usually at higher resolution. We usually have many more synapses or many more elements in the phenotype than we do in the genotype. It also shares in common with generative methods the fact that the same compact encoding with slight changes to the initial conditions can produce different high-dimensional patterns, right? We can change the dimensionality here. We could imagine adding many more synapses in and using exactly the same CPPN. It will just continue to paint however many synapses we have in our neural network. In this case, there's probably on the order of about 100. You can imagine a much more complex robot with millions of synapses. The same compact CPPN is equally capable of labeling all those millions of synapses in that much more complex robot. The question still remains, are those paintings useful? Are they better than the way that we usually do it, which is just take all of those synapses, embed them in one long vector, and let evolution have at those vectors? Like generative methods, obviously I wouldn't be giving you this lead up if this didn't actually work. It tends to actually produce advantageous controllers. Let's have a look at that. We already mentioned this. Why are we going to the effort of evolving neural networks, each of which produces another neural network? We've built in this additional piece of algorithmic complexity to guarantee that all of the neural networks controlling our robots have regular distributions of synaptic weights across their controllers. All good? Okay. Okay, so we want to know whether this actually works, so let's pose a hypothesis that HyperNEAT, the algorithm we just described, can produce better controllers for our robots than the way we usually do things, which in the paper they've given the acronym FTNEAT, or Fixed Topology NEAT. So they're going to do a whole bunch of evolutionary runs using HyperNEAT and a whole bunch of evolutionary runs using FTNEAT, and then give each of those runs the same number of generations and see on average which of these two evolutionary algorithms produces the better robots. So FTNEAT, Fixed Topology NEAT, it's exactly the same as HyperNEAT, except we're throwing away the CPPNs. In this case, we're going to just take all of these synapses, put them in one long vector, and let evolution evolve the elements in that vector. Okay. Which do you think is going to win? Good guess. Okay. Not only does HyperNEAT win, but it trounces Fixed Topology NEAT. So, pretty familiar picture for us here. We have fitness curves. The horizontal axis is the number of generations that elapsed in both evolutionary algorithms. As I mentioned, they're taking the average performance of robots evolved over multiple evolutionary runs using FTNEAT, and then the average performance of neural network controllers for the robots using HyperNEAT. The task here is locomotion, and I think distance here is something like body length, how far the robot actually travels. So we can see that HyperNEAT tends to produce faster moving robots than FTNEAT. Why? We're using HyperNEAT, so we know HyperNEAT produces regular patterns of synaptic weights across the neural network, and this set of results tells us that those robots travel faster. But we're skipping over this middle step. Why do regular patterns of synaptic weights in this neural network produce faster moving robots?
It's because for walking, it's kind of a pattern movement. So if you change how much one leg moves, you should also, if you change how much the mirroring leg moves, then it might be more efficient.
Exactly, right? So if you remember back to our pair of lectures on legged locomotion, one of the hallmarks of legged locomotion is cleanliness of the gait. How clear is the gait cycle? How well do you replicate the same stride every time? Do you repeat it over time using the same foot, like repeating patterns that we see across the body of an organism or in the sine or cos activation function? We want the same leg to do the same motion over and over again, but we also want symmetry. We want whatever the right leg does for the left leg to do that either at the same time or in antiphase with the other. Right? So the Ministry of Silly Walks, one of the reasons why it's funny is, of course, they're inefficient gaits, but they're also not very regular, right? There's a lot of randomness and extraneous motion. We want clean oscillations. And if you can get clean oscillations in the movement of a legged animal or a legged robot, you get faster locomotion. Okay, so to see that, let's have a look at a couple of the solutions here. This one is, we're going to look at, first of all, we're going to look at FTNEAT down here. So this is the case where we're not using HyperNEAT. We're just going to evolve the synaptic weights of this neural network. And we're going to look at the best solution that was found in all the runs that were done using FTNEAT. How's it doing? Not bad. It's a bit of an optimistic simulator. The motors are pretty strong here. Is this a regular gait? Not quite. Almost, right? You can see that 3 of the legs are almost moving in phase with one another, and all 3 are almost in perfect antiphase with the 4th leg, but not quite. You can kind of see the quote-unquote idea that evolution had in mind, right? but didn't quite get there. It was trying to get 3 of the legs to move together and 1 to move in antiphase, wasn't very successful. That's the best that HyperNEAT could come up with. We'll now look at the worst evolved gate from HyperNEAT. So this is a real unfair comparison from HyperNEAT's point of view. When we say the worst here, what we mean is that they did 30 independent evolutionary runs with HyperNEAT, and they took the 30 run champions, the best robot from each of the 30 runs, and across those 30 champions, they took the worst. So it's still good, but it's the worst that they were able to evolve. Much easier for evolution to discover a gate in which all 4 legs move together. Of course, we can't see what's going on inside the neural network, but you can probably make a guess. Tell me about the distribution of synaptic weights inside this robot. How does that neural network that we saw that exists in 3 dimensions, how does it produce in-phase motion of all 4 legs?
They're probably all passing through the same sign function neuron.
Possibly. Other ideas? If all 4 legs are moving together, what can you tell me about the values at the 12 motor neurons here? Remember, the 4 rows in this back plate here correspond to the 4 legs, and each of the 3 values correspond to the 3 joints in each of the 4 legs.
That one looks like each row would be the same.
Right? So we probably have repetitions of sets of synaptic weights that are repeated at each of the 4 rows in at least the back layer. We don't know about the other layers. But if we have the same set of synaptic weights arriving at each row, and assuming that the incoming neuron values are also similar, you're going to tend to get similar values at each of the 4 rows, which corresponds to similar motion in each of the 4 legs, which corresponds to a clean gait, which corresponds to faster movement, right? So we've gone all the way from an evolutionary algorithm that has a particular kind of genotype that biases evolutionary search towards regular structure. And as we saw when we just saw when we apply this now to a robotics problem, that regular neural structure corresponds to better function. So we now have an evolutionary algorithm that's pretty complex, much more complex than a genetic algorithm, but it tends to produce things that look much more like the phenotypes we see in nature. We can see repetitions in space and time. In this case, we're not constructing the anatomy, we're just constructing the neuroanatomy, but we get this repetition in space inside the neural network that corresponds to useful function. Towards the end of this course, we're going to look at evolutionary algorithms that, again, evolve both the synaptic weights of a neural network and the anatomy of the robot. And as you can probably guess, we're going to use HyperNEAT to do so. Okay. Let's just carry on here. Let's do some analysis of some of the evolved patterns we see. In the top row here, this corresponds to 2 different CPPNs. And what's shown in each panel here are the sensor values over time. So if you remember back to the sensors assignment when you were plotting the values of sensors over time, you can see the value of the sensors here. This particular panel here corresponds to the video you just saw, where you can see all of the motors, all of the 4 motors in one of the legs, 4 motors over 4 legs, all moving in phase with one another. Here is another CPPN, which moves all 4 legs almost in phase with one another, but not quite. Here are 2 of the better solutions using fixed topology NEAT, and you can see there's much less regular pattern in time here. There is a less clean oscillation in both of these CPPNs. This plot is kind of interesting. This is something we haven't seen before. This is a mutation plot. Let's look at the left-hand one first. This one corresponds to all of the HyperNEAT runs. And again, on the horizontal axis now, we have the number of generations. And each point in this mutation plot corresponds to a parent-child pair. We're going to ignore crossover for a moment. We're going to look at every parent that produced a child CPPN through mutation. So we have a pair of CPPNs. And the height of that point is the difference in fitness between the parent and the child. A positive value means that the fitness of the child CPPN was higher than the fitness of the parent. And a negative value indicates that the child was worse than the parent. What do you see in this left-hand mutation plot? What can you tell me about the evolutionary dynamics going on in these populations?
The difference between parent and child is a whole lot more widespread.
Absolutely, compared to the one on the right, which is for FT-NEAT. So children tend to be way better or more better or more worse than their parents compared to fixed topology NEAT. What else can we see here?
More of the children were inferior.
More of the children were inferior, right? So NEAT is not a panacea. It's not a solution to all the problems. If you make an alteration to a CPPN, if you alter just one synaptic weight in a CPPN, you're altering the entire pattern, right? And throughout this course, I've been telling you never to touch a phenotype with a sledgehammer. We always want to try and touch it with tweezers. CPPNs break that rule. They often make large changes to the phenotype with just a small change to the genotype. And most of the time, as you would expect, if you take a sledgehammer to something, it's really, really bad. But what is very surprising is this upper part of the panel here. Sometimes the sledgehammer, a mutation to just one part of the CPPN, tends to produce radically better children. How is that possible? How does HyperNEAT make that possible? What property of HyperNEAT makes it possible to make one change to the genotype, which has a global impact on the phenotype, but it's a globally good impact? The child does much better than the parent.
I mean, just the building blocks of symmetry and repetition are good for walking.
Exactly. So if we have a parent which is producing locomotion in which all four legs move together, a mutation produces a child in which all four legs still move together, but they move together at twice the speed. That has the potential to be much, much better. Remember when we first started talking about CPPNs and I painted the left-right gradient for you, we made a change. We still had the gradient, but it was half as much as before. We could have made another mutation that would make twice as much gradient as before. Right? So now we're making alterations not just to the behavior of one joint in the robot, but global alterations to the frequency and the symmetry and repetition of patterns over time. What else can you tell me about these two mutation plots? What happens as evolution proceeds? What happens as you go from left to right in either of these two panels? Yes?
The number of children that are worse than their parents increases.
The number of children that are worse than their parents increases, which is what you'd expect, right? Because the parents, as we go from left to right, are getting better and better. We're getting more and more highly tuned parents. So not surprisingly, if we make alterations to them, it's usually for the worse, right? At the very beginning here, you'll notice that there's small differences between parents and children. Why?
Because the parents might not be optimized at all.
They're mostly random, right? Right at the beginning, and you make random changes to random things, and you get other random things that have about the same fitness. Okay, let's end today with this plot. This shows, this is for the same set of two runs, but now we're plotting the result of crossover, not mutation. This takes a little bit to parse. Again, on the horizontal axis here, we have number of generations, and this is actually a histogram. On the vertical axis, we have the fraction of individuals. At generation 100 here, let's have a look at the three solid lines. The three solid lines correspond to HyperNEAT. The green solid line represents children that had lower fitness than either of their parents. The purple line represents children that had fitness better than both their parents. But you'll notice that the orange solid line is above the other two solid lines. And this solid orange line represents the fraction of children that had fitness between their parents. They were better than one parent, but worse than the other. Most of the children in HyperNEAT that are produced as a result of crossover are like their parents. They have fitness midway between the two fitness values of their two parents. This is showing us that crossover is working, right? We're crossing over two parents and we're getting children that are close to both parents. Although purple down here is what we really want, we want children that are better than both parents. That's still a pretty rare event, especially as we optimize the parents. You can compare that to the 3 dashed lines, which is where we are not using HyperNEAT, and we tend to get children that are much more different from their parents. Crossover is not working as well. We'll stop there. On Thursday, we will switch gears and we will tackle the reality gap. You have a quiz due tonight, and undergraduates, you're tackling assignment 9. Thanks very much.